Welcome to Nikolai's genetics lessons, and today is going to be another video about Manhattan plot interpretation. We are going to talk about today labeling, and before, let me in a few words just explain what we see here for those people who didn't see my previous videos. So here on the x-axis with different colors shown different chromosomes. You see these numbers, they exactly refers to chromosome number 1, 2, 3, and so on. In this case, this plot built for 22 autosomes. On the y-axis, they're going to be minus log 10 of p number. And the smaller p number, the more significant it's going to be, and the higher it's going to be position. Here you see two lines which specify threshold. Below this line shown here with black color is just a noise. In the middle, this can be of some interest, but need to be researched further, and those that is over this threshold line would be a hit. That means that the probability that we got this hit accidentally is going to be extremely small, like 1 in 100 million, that we just got these peaks by pure chance only. As you see, the width of each chromosome is not the same and reduced to the end of this row, because chromosome number 1, 2, and 3, for example, bigger than chromosome 17, 18, 19. So when we build karyotype, human karyotype, we start just with the biggest chromosome and go down to the smallest chromosome. And the smallest is going to be chromosome Y. These gaps here in the middle of the chromosome is where chromosomes has very little genes, and this happens around centromere. Now let's talk about labeling. As you see in this graph, the researcher has labeled the top hits with two pieces of information: the gene name and the RS ID. By the way, all these dots here represent single nucleotide polymorphisms, or SNPs. RS stands for scientific reference number. And as you see, we have here one peak on the chromosome 17, another peak on the chromosome 15. These single nucleotide polymorphisms, actually not only these two, but everything you see here are unique to a certain position on each chromosome and has their own unique number. Consider them as social security numbers, as every American has, and no one else has this number except you. So each single nucleotide polymorphism also has its unique number. In human genome, all chromosomes go into copies which are homologous, so slightly different, again mostly due to these SNPs. And our genome about 3 billion letters long, and we have about 5 million single nucleotide polymorphisms along these 23 pairs of chromosomes. And mostly this is what makes us different from each other. So if you're reading some scientific paper and you see labels like this, what you can do? You can check databases like dbSNP or Open Targets Genetics to see exactly what letter changed. For example, A to G or C to T, and some other variants which are possible. But just by reading label, you cannot tell what kind of single nucleotide mutation it is. So it's not coded here in this unique number. Above RS numbers, you see names of the genes. For example, UNC13C / RAB27A. This suggests that SNP is located between these two genes, or affect both of them. These genes are often involved in how cells release chemicals, vesicle trafficking, and for example, another gene, USP22, this is a specific gene that plays a role in regulating cell growth and cleaning up proteins in the body. It is also important to mention that even though a single position in the DNA, for example, cytosine, can mutate into 3 other things like adenine, guanine, or thymine in the dbSNP database, you can only find one unique number, regardless how many mutations can possible in this single position. There are different visualization tools available, so sometimes you can find plot that looks like this. Not necessary that all the chromosomes going to be in different color. We just need to separate one chromosome from another, so even 2 colors would be enough. And for example, here you see unique database number, and also this is the name of the gene. Unique database number and name of the gene within which this single nucleotide polymorphism is found. As I said earlier, when more than 2 variations exist at one spot, scientists call this multi-allelic SNP. And most SNPs are biallelic, only 2 variations exist of them in the whole population, like A or G. Or some can be found that triallelic or quadratallelic. Even if it is quadratallelic, all 4 bases can be found in certain point of single nucleotide polymorphism. It still keeps the same RS number to avoid confusion. In the early days of genetics, different labs discovered different mutations at the same spot and gave them different names. This created duplicate RS numbers for the same location. However, NCBI, which manages these IDs, performs merges. If they find 2 RS IDs for the same spot pointing to the same coordinate, they retire one and merge it into the other. This is why you will sometimes see a note saying RS123 has been merged into RS456. I hope you have learned today more about labeling of SNPs. And this is all for today. Subscribe and see you in the next video. Goodbye.